In this video, we will take a closer look at a little bit more about prokaryotes, where they live, and what types of relationships they have with other living organisms. So, our first little line here in the text says prokaryotes are ubiquitous. And what this means is that they can be found everywhere. There's not a single place hardly on Earth where we don't find prokaryotes, whether we're talking about the eubacteria or the archaea, we're going to find prokaryotes everywhere. Okay. They're also abundant on the surface of and within the human body. Um, there's a report by the National Institutes of Health that says prokaryotes, especially bacteria, outnumber human cells 10 to 1. So we have a lot of bacteria that live on us and in us. The appearance of bacteria, these prokaryotes, they're vital to the ecosystems. They help form soil. They are important for decomposition. So, in addition to soil formation and decomposition, bacteria are vitally important on the roots of plants. They serve as the basis for, food chains and food webs. They are important in marine environments as well, and freshwater environments. So bacteria can live anywhere, and they're vitally important in those environments where they live. Um, there's some new studies that have been done that show bacteria in the air. We're not 100% sure what roles they play in the atmosphere, but we do know that they live there. Um, Bacteria can be what we refer to as metabolically flexible, in that they can switch their metabolism from one energy source to another depending on, what environment they're in or the availability of their sources. Bacteria are very important in the carbon cycle, the nitrogen cycle. Um, so bacteria, as much as we, give them this bad rap because of the diseases they cause, they're vitally important. There are a lot of researchers who kind of theorize that humans and other organisms couldn't live. We couldn't survive without bacteria. Of course, the other way that they have a direct impact on our health is those pathogenic bacteria. They may be players in climate change. So, bacteria, vitally important for us. Now, when we look at the relationships that living organisms have with one another, this is referred to as symbiosis. And we can have different types of symbiotic relationships, and these are summarized here in table 4.1 for you. So you see there's basically 5 different types of these symbiotic relationships. Our first type is mutualism. And in mutualistic relationships, both organisms benefit. One example that we have that's a mutualistically beneficial relationship with bacteria is with E. coli. Not the E. coli 0157H7 that I was talking about in my last lecture, but just plain Jane E. coli. It lives in our intestines, and it can produce vitamin K and also biotin that are beneficial for us, and then it gets a place to live. So it receives nutrition from us. So both benefit. We benefit because we get the vitamins, and the E. coli benefits because it has a place to live. Amensalism. Amensalism is when one population is harmed and the other is unaffected. So it really could care less. One example of this, or a couple of examples, are some of the bacteria that we have on the surface of our skin, like Staphylococcus epidermidis and Propionibacterium acnes. A lot of other bacteria that live on the skin, um, and like these, they can cause infectious diseases. But as long as they stay on the surface of the skin, then they don't harm us at all. But they produce some bactericidins that can kill other bacteria, that can target pathogens, but um, they're not affected by those at all, and we're not really affected by those. So they can kill other bacterial species, but yeah, they don't really care. They're unaffected. Commensalism is when one organism benefits and the other is unaffected. And the majority of organisms that live on us and in us exhibit a commensalistic relationship. We don't really care. We're not deriving any harm from them. We're not deriving any benefit from them, but they're benefiting because they have a place to live and they have, food to eat. We have a lot of bacteria that live on our surface that digest our dead skin cells. That's not really a benefit to us. Our dead skin cells are going to slough off anyway, but it provides food. These skin cells provide food for the bacteria, so that would be a commensalistic relationship. Neutralism is when there is a relationship, but neither are affected by the relationship. Um, your book provides an example of that one here, is when we have the coexistence of metabolically active bacteria and the endospores, that neither one really cares about the other. This is kind of a newer type of relationship that we've observed. The one that we study the most, of course, is parasitism, because this is when one organism benefits and another is harmed. So when we talk about all of those pathogenic bacteria, this is an example of parasitism, in that they're benefiting, they have a place to live, but we're harmed by that. And we can have different types of parasitism. We can have the pathogenic bacteria, but we can also have, endoparasitism, like with a parasitic worm, for example. We can have ectoparasitism, like with ticks, as an example. But with parasitism, one organism is going to benefit and the other is going to be harmed. Now, the goal of any good parasite is not to kill the host. It can harm the host, but if you kill the host, now you don't have a place to live. So, with parasites, the best parasites, so to speak, are those that can cause harm to their host but not kill their host, because then they have to look for a new place to live. Now, if we take a closer look at the bacteria that live on us and in us, we call that the microbiome. And we call these organisms microbiota. You may hear them referred to as our normal flora. That's kind of an older term. But we have resident microbiota, those that constantly live on us and in us. And then we have transients. Transients are temporarily found. Hygiene and diet can alter our resident and transient microbiota. When we look at our resident microbiota, it's incredibly diverse. We have thousands of different species that live on us and in us. If we think about the mouth, for example, we have a lot of different mutual bacteria and commensal bacteria that live in the mouth. Um, the um, inner surface of the cheek, we know, has the least diverse microbiota because it's exposure to oxygen. But if we start to look at the tongue and in between the spaces in between the teeth, in between the cheek and the teeth, there's less oxygen available there. So we have a lot more different kinds of bacteria that live there, those that can live in the absence of oxygen. Um, we also know that if we were to take samples of the resident microbiota from the mouths of different individuals, we would see incredible diversity there. One thing that is beneficial for us with regards to our residential microbiota is that they can help keep these transients, these pathogens at bay. Our resident microbiota, they have it pretty good. They have this place to live, and they don't want other organisms coming in and taking over their territory. So, by having this wide variety of organisms that live on us and in us, it helps protect us from pathogens. They occupy these niches that would otherwise be open to pathogens. For example, if we think about the vagina, we know that the vagina is full of bacteria. We have resident microbiota that live there, and one of the dominant species is Lactobacillus. And Lactobacillus helps to keep um, the pH of the vagina slightly acidic because it produces lactic acid. And this acidic environment is pretty inhospitable to other bacteria, and it inhibits the growth of yeast as well. However, if this population of lactobacillus changes, or we don't have as many anymore, for instance, if a woman is taking a broad spectrum antibiotic, then it can kill off these lactobacillus species. And now, if there were a few yeasts that were living there, they can start to divide. And if they divide unchecked, then it can lead to a yeast infection, Candidiasis. So, when we disrupt our normal microbiota, then it can allow these pathogens, what we refer to as opportunists, to come in and start to take over. A last little bit about the prokaryotes here. Um, a few words about the archaea. Remember, I mentioned the archaea. These are kind of the extremophiles. They live in these extreme environments. Um, they are different from bacteria, different from the eukaryotes. Their cell membrane is a little bit different. They do not have cell walls with peptidoglycan. We call it pseudopeptidoglycan. And their genomes, meaning their chromosomes, their DNA structure, are larger, they're more complex than those of bacteria. The archaea kind of share characteristics with both the bacteria as well as the eukaryotes. They have some hallmarks of both. Um, they're very complex, and we want to have a way to classify them. Remember, taxonomy, we want to have things classified and organized, but it does make it a little bit more difficult to classify these. We don't typically see these in the human microbiota. None are currently associated with infectious diseases. They play roles in the environment. We have some that metabolize sulfur to produce sulfuric acid. We have some that reduce hydrogen or use carbon dioxide. We have some that produce methane that we call methanogens. So the archaea are a very broad, diverse group of organisms.